I think the intentions behind it are really good. And so I want to, promote those behaviors, but I also want to keep people informed so they're not, spending lots of money on things that are completely invalid, like the IgG food sensitivity test or something like that, trying to look for a problem that isn't there.
Welcome back to the Clinical Athlete Podcast. If you're not familiar with Clinical Athlete, we're a network of healthcare providers, students, and coaches who specialize in the management of athletes. You can find your nearest Clinical Athlete provider at clinicalathlete.com. We also have the Clinical Athlete Forum, where we discuss and share ideas and resources related to athlete health and performance. To join the forum or for potential listing on the Clinical Athlete Directory and for all upcoming seminars, webinars, and events, details can be found on the website. This podcast can also be found on your favorite platform, and if that platform allows you to rate the show, we'd appreciate you taking the time to do that so that we can get this information out to as many people as possible. My name is Quinn. I'm a doctor of physical therapy in Orange County, California at Clinical Athlete Newport. On this show, we are joined by co-host Jared Maynard, who is the Clinical Athlete Continuing Education Director and Coordinator and a physiotherapist at Depth Physiotherapy in Waterloo, Ontario, Canada. He is also a strength coach and runs an online powerlifting coaching company and is a competitive powerlifter himself. And we have our other co-host, John Flagg, who is an athletic trainer and the powerlifting, weightlifting, and strongman coach at 301 Strong in White Plains, Maryland, and the owner of Rebuild Stronger, an online coaching platform for strength athletes. He is also a clinical athlete provider and lead instructor of the Clinical Athlete Powerlifting Certification. And we're very excited to welcome onto the show Dr. Gabrielle Fundaro, who has a PhD in human nutrition, foods, and exercise with a special interest in the gut microbiome. And that's what we're talking about on this show. Dr. Fundaro dispels some myths and drops a ton of knowledge on a subject that we've not yet talked about on the show. And who doesn't love a good discussion about or related to poop? Hope you enjoy the show.
Welcome back to the Clinical Athlete Podcast, everybody. This is John Flagg. I'm a certified athletic trainer. And today we have Quinn from the land of butterflies. Say hi, right. Quinn.
That's California, for those of... that's like a year old now. I don't think that... We had that butterfly influx last year.
Still relevant.
Okay, fine.
I like it.
I'm just the butterfly guy from now on.
That's what you're going to be.
Fine.
And then Jared in the land of snow beasts and hockey.
Absolutely. Canada land.
How you doing, buddy?
Wow.
I'm good. Happy to be here as always.
Cool. So today it's a bit of a different topic. We do a lot of rehab and clinical stuff. But I first met Dr. Fundaro at the RP Summit, and she talked about the gut microbiome, and it was incredibly compelling to me. And we see it's such a hot topic on social media, and I know I get a question a week about gut health and performance and how people can impact that. So I thought it'd be great to bring her on. Dr. Fundaro, why don't you tell us a little bit about yourself, how you got started in the field, and anything else that's interesting.
Okay. Well, thank you for having me on. And I am talking to folks from the land of the sweaty armpit of the United States, aka Georgia. No, Georgia. I'm in the Southeast. It's just swampy down here. So yeah, I bounce around quite a lot, but this is where I've been for a little while now.
Setting the stage for the conversation.
We're going to talk about poop and sweaty armpits, got to be on the same page. So I started in the area of gut microbiome before it was cool. So I guess I can be like a gut microbiome hipster. I don't like to, people are like expert. I'm like, no, not really. No one knows enough yet, but I'll be the gut microbiome hipster because I studied this for my PhD and I finished that back in 2014. So it was sort of a happy accident that brought me into the world of the gut microbiome. I was initially studying the effects of high-fat feeding on muscle hypertrophy or lack thereof, so cachexia or muscle wasting. And I had a side project looking at probiotic supplementation and its potential protective role during high fat feeding. And the way that became my main project was that we had some accidents with sample prep in the lab. And so I lost all of the samples from my main project that we needed. And that was after months. We had so many cohorts of mice and everything else went off without a hitch. I mean, could not have been a more perfect I can't make words. Experimental protocol and data collection and sample collection. But of course, it happens. That's research. So fortunately, we had the side project, and that became my main project. So we put multiple cohorts of mice through this study, and it looked a lot more like, instead of like cool, me in a lab coat, like pipetting stuff, I was tube feeding mice every day, 7 days a week for like a year. So that's what probiotics research looks like sometimes, and collecting a lot of poop, and a lot of human poop too. So we were looking at the, as I mentioned, the potential protective role of probiotic supplementation during high fat feeding based on the theoretical mechanism that some of the metabolic dysregulation that we see during high fat feeding is regulated by compounds that come from the gut. And so there's this idea that you could potentially have a gut that predisposes you to obesity or metabolic dysregulation or a host of other problems or potential benefits. And so via, probiotic supplementation, maybe we can correct that or attenuate it in some way. And I came out of that program right into an assistant professorship in exercise science, and I really didn't intend to do much else with it. I knew that I wanted to teach from about the age of 20. And so I was like, okay, next step is PhD. And so I went, straight from bachelor's to PhD, do not pass go, do not collect $200. And so I came out of that program fairly young and worked in the exercise science program for about 4 years. And I was primarily teaching sport nutrition and anatomy and phys, which, is the really horrible weed out class. And those professors are always the absolute toughest and worst. And I totally fit that stereotype. I was super, super tough. But Mike Israetel of Renaissance Periodization stumbled upon me in the International Society of Sport Nutrition Facebook group. And I had like no social media presence. I had like a little blog, and I was just this little power lifter. Like, this is my pre-workout supplement that I make myself. And, here's my safe weight cut protocol, and, posting in the ISSN. And I had, a debate, I guess, with someone on that page, and he, followed that and appreciated my content and the way that I carried myself, I suppose. And so he recruited me to Renaissance Periodization. So for a year, I was, full-time professor and then also coaching on the side. And I did a few seminars that were not really RP associated, but because people knew that I was an RP coach, it gave me a little bit more, I guess, leverage in that arena. And after one of them, I was like, man, this is really awesome. I get to educate folks who are really engaged in the material, and there's, such a great dynamic here, and people are excited about this. And I really seem to be able to reach people and my clients. It's such fulfilling work. And not to say that academia, is not ever like that. It can be, but I just felt like I was being pulled in another direction. And so I ended up, after a year, resigning from my professorship and went right 100% into coaching, which was kind of a scary move at the time, because you go from having, a salary and health insurance and benefits and whatnot, retirement plan, to, going out on your own into the wild west of the fitness industry and being an entrepreneur, which I am not. Like, I'm not a business person in any way. And I would do this all for free if that, didn't leave me starving to death living in a box. So yeah, that's how I got into this. And when I resigned and, was going in full time, Mike and I talked about this gap in the industry where we have, a lot of emerging data about the gut microbiome, and it's very exciting and there's so much potential here. But because there's such a gray area and maybe some difficulty communicating the literature, to the people who want to be able to apply it, there's a lot of misinformation, and marketers are sort of capitalizing on that misunderstanding and using it against people to make money off of them. And so that's kind of where I stepped in to be as best as I can an evidence-based voice and sort of a voice of reason and kind of disseminating and translating the literature, to the greatest of my ability so that people can be informed and empowered with it rather than being taken advantage of.
Yeah. And that at the summit, I think you probably got pelted with the most questions when it came to the gut microbiome. And some of them were Facebook clicky article type of questions, is this going to fix everything? A question that I have queued up here is, if I get this right, will it make me immortal? People believe it's like the panacea right now. And in at least my understanding of it, that's not, we're not there yet. We don't have that kind of information. So I really want to kind of frame this discussion, and I want to start with just kind of the basics for people so we can outline and get them a good little foundation to start with. So really, what is the microbiome? And one thing that you mentioned during your talk that I didn't know is that it actually comes in different sections. Everybody thinks gut health. You have a biome in your large intestine and throughout the entire digestive process. So can you outline that for us a little bit and give us a frame of reference to start?
Yeah. And I want to say one thing about the seminar, which I thought was sort of like tragically funny, but I got, we all got feedback afterwards, and some of the feedback was like, well, we didn't get what to do. Like, you didn't tell us what to do about this. I didn't get like a practical takeaway for like, what are the interventions? And it was like, I know, but that's because we're not there yet. So yeah, exactly. It's kind of one of those things that I'm like, okay, here's what the data says so far. Like, do with this what you will, which is probably not a lot right now. So I love that you said that. Yeah, the science is just not there yet. So yeah, and I think that was that's one that people are kind of surprised to hear. And something that I really stress is that when we talk about the gut microbiome, it's not just one static object that looks the same from, mouth to anus. It's very different. And we even have, the oral microbiome being separate. So when we're saying words like the microbiome or the microbiota, that refers to all of the microorganisms that would inhabit the area that we're talking about. We have more than just the gut microbiome. And then the microbiome is referring to all of the genetic material. So we have the organisms themselves, and we have all of the genes that they could potentially express. And so genes being sort of the recipe books for proteins, and each of those proteins would serve a different function. So we actually have an oral microbiome, we have a skin microbiome, women have a vaginal microbiome, and then we have the gastrointestinal microbiome running from stomach, small intestine, and large intestine. And then what we usually use in literature to sort of extrapolate and assume some things about what's going on in the gut, we have the fecal microbiome. And so each of those populations looks a little bit different due to the environment. So we can think of our stomach. Most of us know that's super acidic. It's one of the first lines of defense in fighting off pathogens that we might ingest. So there's a low abundance and a low diversity of microbes because most of them don't do well in a very acidic environment. And then we have the small intestine, and we divide that up into 3 sections as well. We have the duodenum, jejunum, and ileum. So the duodenum is closest to the stomach. It's still fairly acidic in there. So again, we have fairly low bacterial abundance and diversity. As we move to the more distal end of the small intestine, we start to see that we have less oxygen and less acidity. And so bacterial numbers start to climb a little bit and we have a little bit greater diversity. But where we really see a bloom of abundance and diversity is in the large intestine. So where the small intestine meets the large intestine, that's called the cecum, and that's sort of a hotbed of bacterial proliferation. And then throughout the large intestine and in the colon, or in the distal part of the colon, near the rectum and the anus, that's where we see the greatest abundance and diversity of bacteria, and all microbes, really. And that's the other caveat. This is not just bacteria. It's about 99% bacteria, but we also have some other organisms in there. We've got archaea, we've got fungi, including yeasts. And a lot of people are like, oh, yeasts are so bad. No, they actually play an important role in the gut as well. The archaea play a really important functional role in helping to maintain the fermentative capacity of the gut. And then we also have viruses there. So they're non-living, but they do play a role in shaping the population because they can infect human cells or bacterial cells. So the majority, about 97 to 99% of our gut microbiome is located in the large intestine. And altogether, all of those microbes are a couple pounds of biomass. So we do have a huge amount of just a huge number of bacterial cells. So the number of bacterial cells that we have are about equal to the number of human cells that we have in the body. But the genetic material there outnumbers ours by thousands of times. So much genetic material. So for us to kind of make statements about, ways that we can influence the microbiome, especially when we're, talking about like remodeling it in some way, I think we're really taking it a step too far because there's just so much there that we haven't figured out yet at all. And they are, so we, they do play an important role also in human physiology. So as I mentioned, defense against pathogens. They help to mature our immune system. So that's from really, upon the time of birth all the way through adulthood and throughout our lives. They modulate our immune system as well. They can produce some nutrients. They can ferment fibers to short chain fatty acids, and we can use those. And that's actually kind of a big topic in the area of exercise as well. And they sort of fight with each other as well. They're constantly vying for nutrients and for real estate. And so they're interacting with each other. They're interacting with our cells. So it is a really complex population, sort of another ecosystem, we could think about it in that way, existing within our bodies.
And this is really that beneficial type host scenario. And that's it. You see it in the literature a lot. And it took me a little bit to get used to see host as the subject. It's like, oh, this is the kind of relationship we're talking about here. Do you want to touch a little bit on diversity and its positive role and like what that really means when it comes to the microbiome?
Yeah. So diversity, we can look at it in a few different ways, but it generally refers to the number of species present and then how their relative abundance. So, are we like 60% of one and 40% of another? We can also look at them in terms of their relatedness. So just like on a family tree, are they very similar? Are they dissimilar? We can also look at diversity in terms of the functional diversity. So not just who's there, but also what are they doing? So we can look at gene richness as well as species richness. And what's really interesting is that when we look at diversity and we look at sort of the, if you could think of this like a snapshot, a family photo of your gut, and you're a healthy person in the US, your family photo will look very different from the family photo of a person who's in Korea or West Africa or South America. And so what the problem that poses then is that we don't have a specific profile for healthy gut. We have some key taxa, some key groups of bacteria that we know should probably be there. Some of these keystone species that we generally find in relatively high abundance in humans, that they fill important niches that, we, for example, like digestive enzymes that we don't produce, they will produce for us. So we can outsource some of these jobs to them. And so we've co-evolved with them for a very long period of time. So when we talk about something like dysbiosis or the unhealthy gut, the definition is really muddy, and it's really just any difference compared to the healthy control. And I have to add, it's to the healthy control in that study. because the greatest variability that we see is not due to diet, exercise, disease. It's due to just inter-individual variability. So just one person is so different from another person, even though we have this sort of core group of bacteria and organisms that should be there. About 2/3 of it is just specific to you as an individual. So it's very difficult to characterize a healthy gut or an unhealthy / dysbiotic gut. And even within the same disease, we don't see the same patterns emerging every time. So what we can say at best is that when we're talking about diversity, even just looking at who's there doesn't give us a great picture. And instead, it's probably more important to look at what they can do, because that's one thing that does seem to be emerging fairly consistently, is that in individuals who have a disease or in individuals who have obesity, that it's not necessarily who's there that changes consistently each time, but we tend to see a reduction in gene richness. So that means there's reduced functional capacity or a reduced expression. So they're not, even if the genes are there, they're not being expressed. So that may be more characteristic. Like the true dysbiosis may not be in just the taxonomy, but in the functionality of the gut. This makes a lot of sense because when we're looking at sort of the contribution of different microbes to the overall gut functionality, in some cases we see that those with even very low relative abundance, like some of the archaea, that comprise less than 1% of all of the organisms present, that they play a huge role in the fermentative capacity of the gut. So without those guys, even though they're very low in number, the entire functionality of the gut would be significantly affected. And so I think it is important that we keep that in mind, that even if we're changing the relative abundance of species, we don't necessarily know that we're changing the function in an appreciable way. And that concept is fairly new, and they've called that functional convergence. So even if we're looking at two people who have very different taxonomic profiles, the function will still be fairly similar. So we just conserve those across time, which is good, because it would be really problematic if you were to lose just one group of bacteria and then the whole thing goes to pot and nothing works anymore.
Yeah, that would suck.
I've got a couple questions, and I don't know if they're related at all, but almost taking a step back and defining dysbiosis, or at least maybe defining it physiologically, and also how that manifests, or how we would identify that clinically.
Oh, yeah. Yeah.
Okay, I'll stop there then.
That's a very good question. So, as I mentioned, the definition of dysbiosis is really just any difference compared to the healthy controls. So dysbiosis could be an overabundance of potentially pathogenic species. It could be a lower representation of potentially beneficial species, or it could be a little bit of both. And how that might manifest in clinical practice, well, what's really interesting is that we don't really. So if you were to look, I'll start with the gut. So just as a caveat, though, the preview to what I'm going to say, there probably will not be symptoms that you could identify clinically, from how a person is feeling. So if we look at the gut itself, we can look at the anatomy and physiology of the gastrointestinal tract. So do we have healthy villi, so all of the folds that we see in the small intestine to increase surface area and absorption? Do we have the mucus layer? So we should have a double mucus layer in the large intestine. And do we have intestinal cells that are held tightly together via tight junction proteins? So those are some of the characteristic things that one might look at. So you could have damage to the epithelial wall that causes low villus height or damage to those cells. You could have a reduction in the thickness of that mucus layer, and that means that there's less protection, there's less space between the microbes in the lumen and the intestinal cells and the immune cells beneath. And you may also see either a downregulation or destruction of those tight junction proteins. So the cells are not held tightly together and substances can pass through them. And we call that intestinal permeability or leaky gut. Now, you can indirectly or directly measure some of these things. In humans, we're generally indirectly measuring intestinal permeability. And there are a few different ways we can do that. You could look at fecal zonulin. So that's a protein that downregulates tight junction proteins. So you could look at that as a marker. You could use a dual sugar test. So you have a person ingest two different types of sugars, a mono and a disaccharide, and then you measure clearance into the blood, and that can give you an idea of small intestine or large intestine permeability, depending on the sugars that you're using. You could also look at some markers that might be associated with a disease state, like fecal calprotectin is one of the things that you would measure to look for the presence of inflammatory bowel disease. Now, quite often, these markers of intestinal permeability may correlate with gastrointestinal distress in athletes as part of exercise-induced gastrointestinal syndrome. But in folks who are just not active to the extent that it would cause any intestinal permeability, you're not really going to have any outward signs of intestinal permeability. A lot of the things you would think might correlate with it, like people talk about, gas, bloating, diarrhea, something like that. Those markers are actually not associated with intestinal permeability. They may happen at the same time. They may not. There's no strong correlation between those. But there are some risks for intestinal permeability. And those would include the presence of type 2 diabetes or gestational diabetes, BMI above 25, eating a standard American diet. So those things may increase the odds of you experiencing intestinal permeability, but you still would not have outward symptoms of the intestinal permeability. Now, intestinal permeability is something even that's separate from dysbiosis. And how do we know if we have dysbiosis? Well, that becomes really, really difficult to determine. How could we see who's in the gut? Well, what people most of the time do is take a stool test and they analyze that. Now, of course, there are some fun, over the counter, GI map tests and things like that aren't necessarily, validated. They're not clinical tests. They're giving you an idea of the abundance and representation of species in your feces. But drawing from, the knowledge of the gut microbiome is not static from one end of the GI tract to the other. And that quite often in those fecal samples, we're seeing just luminal contents. That means that we're missing a large portion that we can't quantify, but we know that we're missing out on a large portion of the population there, because there are bacteria that are mucosal associated. So they're actually hanging out right in that mucosa, right in that protective mucus layer, the top layer, and probably more closely associating with our intestinal cells and our immune cells. So we really want to know what's going on with those guys as well. And we're really missing out on that. So these fecal samples are just, they're really a separate sample from what we would get if we went and did a luminal mucosal sort of sample, which can be done, but it's just very invasive because now you're talking about, colonoscopy type procedures to go in there and take samples from the lumen, the mucosa, or the cecum. But they have done that in humans, and so it is possible. But again, it's just very invasive and it's not too common. Now, once again, you're not going to have necessarily outward symptoms of dysbiosis, because dysbiosis and the microbes that we're looking at those haven't been causally linked to any disease outcome. It's just that we tend to see that in individuals who have a disease, they look different from the individuals who don't have that disease, but we're not able to then say, oh, it's this microbe that caused this disease. It's usually just, well, we tend not to see so many of these, and we probably should, but we have to be very careful about developing causation when there's only a correlation. So, unfortunately, at this point, we don't really have clinical tests that we could use to sort of diagnose dysbiosis. It's not a diagnosis at this point. And even though we can, undergo indirect markers of intestinal permeability, that again is not something that will necessarily have clinical relevance. It's something that can certainly occur after a bout of exercise, and that's probably the most applicable to your audience. And there are ways to go about managing that, but it's not super sexy. I'll say that ahead of time. These recommendations are always like, oh, it's just pretty much the basics. I'm like, yeah, that's where we are right now.
I think some of the causation point that you bring up adds a whole lot of confusion, because if you see some type of disease process and you're like, oh, this is also correlated with a altered microbiome, then when we retest, let's say that disease process is gone, does the microbiome then normalize? Is there any data on that where they're looking at disease processes from start to finish?
So there are changes to the profile, for example, in individuals who have an inflammatory bowel disease, in those who are in an active flare versus in remission. And there have been studies looked at changes in response to dietary interventions in individuals with obesity. So the diet itself has an independent effect. And so you can see changes to the microbiome in response to the diet before weight loss has occurred. But the thing is, we don't know, necessarily what the clinical outcomes would be from those changes. And we have this chicken versus egg problem where we don't know if the disease, causes the changes to the microbiome or the microbiome progresses disease states. But it does, I think that we're starting to, because we keep seeing patterns emerge with just very specific taxa, that I think we're at least starting to say, okay, probably like these groups, still not a causative, but maybe it gives us a little bit more of a clue for a potential intervention. So like Akkermansia mucinophilia is one that we tend to see change pretty consistently with various diseases. And it's one that's considered to be beneficial. Or with, long-term fiber deficient diets that happen to be associated with deleterious health outcomes, we tend to see reductions in bifidobacteria, and that's another beneficial one. So I think that it's emerging. We may be able to get to that point eventually, but yeah, it is. It's really just a chicken versus egg problem. And even if we do see changes to the microbiome, we don't know necessarily that it's just because of the disease regression or improvement, because we also have to look at what did we do to help improve, the progression of this disease or to resolve it.
To jump in on that point, how well do you think that is conveyed, or the accuracy of those points are conveyed in, say mainstream media outlets when it comes to these sorts of topics? Because, I can think of some family members who tend to like to watch documentaries on TV about this stuff. And It's been a long time since I've seen one, so I don't know if they're saying these things are correlated or if they're saying this causes that disease. But it seems like the effect at the end of it is still like, well, the microbiome is linked to that thing, so we need to do X, Y, and Z to prevent that thing. So, with your position in this field, do you get a lot of people coming to you saying, hey, I heard that the microbiome functioning this way leads to this disease process? And then how easy or difficult do you find that it is to have conversations with those people to try to say it's not nearly that cut and dry as of right now?
Oh man, that brings to mind a recent headline I saw. Your gut microbiome predicts how likely you are to die in the next 15 years.
Oh.
And I was like, no, No, it doesn't. We're not there yet. I reading I have to go and, read the actual article, the primary source first, but I just went and read, just the media article because I was so curious. And it was something like, they were looking at all of these other factors and, predicting like death in the next 15 years or like looking forward, who dies in the next 15 years. And it was 15% more accurate than a bunch of these other measures. But it was like, okay, 15% more accurate on top of something that was probably not very accurate to begin with. That doesn't mean that your microbiome, like, you go get your GI map test and it's like, oh, you got 10 years left, but like that's the way that it's promoted. And I think also, to be fair, like people want interventions, which is totally understandable. They want to know that this is something that they can exert control over in some way. And I think what we really lose sight of also is that, there are certainly interventions that have a measurable and reproducible effect on the gut. But we also have to look at the magnitude of effect. So if we're talking about thousands of species and we influence 2 of them, that might be really cool. It might mean something, but it could also mean that, we're only influencing those 2 species. And then as a whole, the ecosystem itself stays relatively stable. And that's what we really see in longitudinal studies, that for the most part, regardless of what you do, things stay stable. And even if you do undergo some sort of dietary exercise intervention, if you stop that, obviously things will go back to baseline. But sometimes, even after months of that intervention, things still go back to baseline. So, the extent to which we can actually exert any influence on that population is still, I think, remains to be seen. And at this point seems pretty minimal. But it just so happens that a lot of the things that people want to do in order to shape their microbiome, they tend to be healthful things. So that's great. I'm like, if you want to now suddenly eat a lot of vegetables and, partake in more physical activity, like, okay, yeah, let's do that for the sake of your gut microbiome. And like, also you'll just by accident eating more vegetables and, exercising more. So it's like I think the intentions behind it are really good. And so I want to, promote those behaviors, but I also want to keep people informed. So they're not, spending lots of money on things that are completely invalid, like the IgG food sensitivity test or something like that, trying to look for a problem that isn't there.
Hey guys, Quinn here. Consider this a little brain break from our really interesting discussion with Dr. Gabrielle Fundaro about the gut microbiome and other poop-related concepts. You see, I'm just taking every opportunity to say the word poop on the show. You've probably heard, but just in case you haven't, Clinical Athlete is teaming up with the Level Up Initiative and putting together the first ever CALU Summit on September 19th, 2020 in Boston, Massachusetts. We decided to come together on this because, let's face it, clinical practice is hard, and conflicting information can leave you feeling lost. With the endless amount of research and dogmas, clinicians may feel frustrated and confused about how to help their patients. The CALU Summit is your solution to help you gain confidence and clarity with key rehab principles, including exercise prescription, pain science, and communication. We're centering these concepts around 3 common clinical cases that you may encounter: ACL reconstruction, low back pain, and tendinopathy. Gain confidence in your clinical practice and find your path to success. And hang out with a bunch of badass providers from our communities. For more information on the summit, head over to calusummit.com. That's CALU summit.com. You can also find the link in the show notes. Hope to see you there. And now back to the show.
I think it's a good time here to also take a little bit of scientific responsibility and explain, like you mentioned, you saw that article and you're going to chase down the primary article. And there are times where I've seen, study says, and you go to the primary article and it's about mice getting like a fecal transplant that you had mentioned before. Like, is that necessarily applicable to humans? Have we been able to reproduce it? And we need to be really careful extrapolating that out into a different population. Like, I'm sure you've probably seen that quite a few times. Are there other examples that you can think of there?
Oh, yeah. Yeah. Artificial sweeteners for sure. If you want to show that artificial sweeteners are really harmful, do your test in cell culture. If you want to look for, mechanistic data, then you might use a rodent model because the benefit. So, a rodent gastrointestinal tract is very different from a human gastrointestinal tract. And even if we use, specific, I can't think of the word now, but when we take a fecal transplant from a human and we put that into a mouse and we give that mouse a human-like gut microbiome, the relative abundance is still not the same. It's not equivalent to a human microbiome because these bacteria rely on the environment, and the environment in a mouse gut is different from what we see in a human gut. So we can use that for mechanistic data, though, to some extent, because we can sacrifice those mice at the end of it and take out sections of their gastrointestinal tract and take out their skeletal muscle and look at things, from a cell signaling angle. But if you want to see what effect something will have in humans, you really have to do it in humans. The closest surrogate would be pigs. So we do have, porcine studies are not as common because it's very expensive and whatnot, but that's kind of the second best. Their gastrointestinal tracts are very close to humans. But, the amount of rodent data that we have far outweighs that of what we have in pigs or even in humans. But yeah, so with artificial sweeteners, in cells, significant negative effects, significantly damaging effects. In rodents, we can give them super physiological doses and still exert some pretty negative effects and show that, wow, artificial sweeteners are really bad for you and can cause cancer and seem to be really bad for the microbiota as well. And then when you take that out into humans, at doses that are physiologically relevant, well, then we usually don't see much of anything. But that being said, there have been some studies that have shown some things in humans, yes, but then we have to look at things like study design. Well, if there is no control group, or you had a very small N size, you had 3 or 4 individuals, we can't then really readily extrapolate that to, the rest of humanity. So we always have to keep that in mind, that we're looking at, just samples. And what I find really funny is that, we will have these huge meta-analyses that have tens of thousands of participants, and people will be like, well, I don't know if I really buy that. These studies are funded. And then you get one study on artificial sweeteners and 3 people, and people are like, I knew artificial sweeteners were evil. And I'm like, oh, the cognitive dissonance there. So I try to be very, pragmatic and, cautious in my recommendations and my statements, and try to deliver them with context. Even if sometimes I get excited about stuff, and I know that I have my biases for, I'm big into vegetables and fiber. But I still try to keep an open mind, and I look at, studies that might contradict what I think, and, talks that might contradict what I think, because, I believe that it's important to be able to argue for both sides. Keep your mind open, but not so open that stuff falls out.
Yeah.
Going back to that article that you read on, my mind is blanking. What was you read the media article?
Yes, 15%.
Yes.
I can tell if your microbiome tells you you're going to die in 15 years.
We're getting nocebo through our poop.
Sorry. So yeah, was there a yogurt ad at the bottom of that article? So, and I'm joking, but my next question asks, what are probiotics? And well, we can go there. We can just stop there. But what do they do? What do they not do? Because I hear that term is thrown around.
Oh yeah.
In this, as far as this realm, probably more than any other that I hear.
Yes. Yeah, absolutely. Because probiotics are sort of, that it's if you want to get a targeted attack, you want to change the bacteria, you got to eat the bacteria, and then, you get some magic out of that. So probiotics. So we've got probiotics, prebiotics, symbiotics, and postbiotics because we just keep adding more biotics.
And antibiotics? No, that's different.
And antibiotics. Okay. So kind of, yeah, it's related. So a probiotic is a microorganism that you ingest, which confers some benefit to the host. It used to be that they were identified as live microorganisms. Now we know they don't necessarily have to be alive to exert some effect. So they can be heat killed. We also now have spore forming probiotics. So these are from bacteria that exist in sort of a dormant state. They have great survivability through the highly acidic stomach, and they can exert some effect once they reach the intestines. So that's probiotic, organism that you ingest. Prebiotic is a nutrient source for the organisms. And those organisms may be the ones that are already in your gut, the commensals, or, if you wanted to take a probiotic organism, that could certainly utilize the prebiotic energy as well. And so these prebiotics really could be any nutrient that passes through the small intestine and reaches the large intestine. But most often we're talking about microbe accessible carbohydrates. So these are mostly fibers that we cannot digest. And because they're not accessible to us in the small intestine, they reach the large intestine where they can be fermented, really releasing energy for the bacteria, and then potentially short chain fatty acids for us. And then we have symbiotics. So that's when you take a pre and a probiotic together and they formulate these. They'll have like an inulin or something in them. And then postbiotics are the products of the bacterial fermentation that confer some benefit to the host. So an example of that would be the short chain fatty acids like butyrate is the postbiotic. You can take butyrate, you can take propionate as supplements. You also find them naturally occurring as short chain fatty acids in the diet. So those are the big iotic words. And then obviously we have antibiotics used to kill bacteria. And those are actually produced by bacteria also. So bacteria can produce antimicrobial compounds to control each other's numbers as well. So probiotics, that is, and if I feel like if anyone should be like, yeah, probiotics, maybe it should be me because that's what I did in my for my dissertation was study probiotics. And like I was really interested in them. And I thought, wow, this is an area like that seems to have so much opportunity and potential for, interventions and improving human health and whatnot. Not to say that they don't have applications. We do have some pretty compelling data in certain areas, but the realm of probiotics research is so problematic for a few reasons. One, pretty much every study is done with a different methodology. So you have different strains of bacteria being fed to different populations in differing amounts for different reasons. Whereas if we're looking at something like creatine supplementation, like we've got lots of reproduced creatine supplementation data in similar populations with similar effects. So we can be really confident about like, when do you take it? How much do you take it? What can you expect from it? And we just haven't really been able to do that with a lot of probiotics. The other thing is the effects of probiotics are strain specific. So when we look at, how we classify organisms in terms of taxonomy, we used to really talk more at the phyla level. So it was looking at like vertebrates versus invertebrates, Bacteroidetes versus Firmicutes, so general. And we now know that the rate, we used to say, oh, your beta-F ratio, that really predicts like obesity propensity. Like not really. And then we can get down to the genus and species level. And that's pretty similar. So now we're looking at like canids, like dogs, foxes, jackals, dingoes, and we can get more and more specific down to species, dogs versus dingoes. This is the one they use all the time because everyone knows what a dog is and everyone knows what a dingo is. And like, yeah, I think that like some people can domesticate dingoes, but like for the most, like, for like the standard American house pet, like you're probably going to want to pick a dog. And then, not everyone needs to have a dog. Not everyone needs the same type of dog. So it's the same thing with probiotics. The effects are strain specific. So it's not uncommon that you might see a strain like Lactobacillus rhamnosus GG. This strain seems to be very effective for pediatric diarrhea. So like if you have there's replicable ped studies looking at, this is really helpful for kids who have diarrhea. Super. But if you give it to like an athlete or any other adult, it does like nothing. So, do we say that probiotic is not effective? No, it has effects, but in a specific population for a specific thing. And it's the same thing when we're looking at Saccharomyces. So it's a yeast that seems to be consistently beneficial for adults with diarrhea associated with antibiotics or traveling. And there are some other strains of lactobacillus and bifidobacteria like brevis and animalis. Those seem to be consistently beneficial for some things. And then, in the realm of athletes, this is another one where we don't have a lot of evidence and a lot of strong evidence. And unfortunately, where we do have replicated studies, sometimes they're just from the same lab. And I think that it's important that other labs are also able to replicate those studies. So we can't say that there's one that's 2 studies have shown reductions in delayed onset muscle soreness. Well, it's from the same lab. So if we're looking at the same lab with the same, population, can we really say that that's 2 pieces of evidence? I don't know. I guess that I leave that up to people how they want to use it. But I tend to be very skeptical. So when we're looking at, where can we use probiotic supplementation? Pretty much if you have diarrhea, if you have an inflammatory bowel disease, if you have irritable bowel syndrome, or if you're a child and you have diarrhea, there's probably a probiotic strain that you could use in conjunction with other lifestyle things that might help to reduce your symptoms. Or if you're an athlete, there are some strains that seem to be helpful for reducing the incidence of upper respiratory tract infection. But aside from that, how do people really want to use them? To lose weight, to perform better, for mood. We really just don't have the evidence, or we have evidence that they're not effective for those things. And then the other issue is that when we look at meta-analyses and systematic reviews, sometimes they'll say yes, probiotics were effective for this thing, but they're looking at a bunch of different probiotics and some of them weren't effective. So it's if we were to just say do sports supplements improve performance? And as a whole, a lot of them on the market don't. So we might say no, and then miss out on the fact actually creatine is pretty effective. Caffeine's pretty effective. So it really also depends on what we're including in that analysis. And it's just hard to do that. So that's why I think it's important to, I always fall back on those larger analyses. But then if I find something that I'm this is curious, this is unusual, or, I really want to pare down, then, go down the realm and find the individual randomized control trials and see if you can paint yourself a clearer picture of what's going on.
Well, that probably leads us into the unsexy actual recommendations, because it looked like you gave us a lot to read. You did. And,
I know.
But it's fine. I actually really liked it. And I started with the Monda paper, the review article. And if anybody is listening and wants to know, that was exercise modifies the gut microbiota with positive health effects. And it was a review article. And just the way it was written, it was much more digestible for me. That's how I know I'm a dad. I just hit you with a dad joke and I didn't even realize it was coming out.
Yeah, that's smooth.
Sorry.
You're not, though.
Wow. I'm not at all. Not at all. But it seemed like if you look at a lot of the reviews that a healthful diet and regular exercise seem to be pretty good indicators of diversity and all the things that we at least link with a positive kind of microbiome. Is that kind of what a lot of the evidence is showing, and what the suggestion would be? Is just, hey, diet and exercise.
Oh yeah, absolutely. That's what it is, because we can pretty confidently say that physical fitness level, and they are generally using VO2 max. So they're looking at cardiovascular fitness tends to correlate with increased microbial diversity and specifically enrichment of butyrate producers, and just that from, the fermentation capacity of the gut. So how many of these do we have? And then how much butyrate can we actually produce? And then butyrate has a number of positive health benefits, helps with insulin sensitivity, appetite regulation, tight junction protein regulation. So butyrate itself seems to be really beneficial. So yeah, increased levels of cardiovascular fitness, probably a good thing. Athletes tend to also have kind of just higher energy availability, and they postulate that maybe it could also be due to increased carbohydrate intake, that they tend to see higher levels of some of these butyrate producers, bifidobacteria. Prevotella was really big a couple years ago. Now we've got this Veillonella, who appears to consume lactate as its sole carbon source, and it spits out propionate. And if you give a mouse a propionate enema, it improves its exercise performance. So just basically, some of the effects of exercise on the microbiome and vice versa, that seems to have sort of a reciprocal relationship. And so we see benefits from physical activity. And then we also see benefits from having a diet with plenty of microbe accessible carbohydrates. Now, proponents of the carnivore diet will say, oh, well, we don't need fiber, or fiber in diverticulitis, because they, incorrectly assume causation where there's correlation. So can we make some of these short chain fatty acids from animal products, from amino acids? Yes. But this is another thing that's pretty consistently shown, that if you have a diet that does not have enough fiber, that you may see a thinning of the mucus layer, and that you'll generally see reductions in microbial diversity. And even in athletes, in bodybuilders and cross-country runners who are physically active, a really low fiber, high fat diet doesn't seem to overcome the benefits of exercise. So basically, they say you can't outrun a bad diet. Like, you can't outtrain a lack of fiber. You still need the fiber. You still need the substrates for those bacteria. And then, one area that I think a lot of people would struggle with is in endurance athletes, that they're experiencing pretty severe GI distress. And that's sort of a special group. If you have athletes who are endurance or ultra endurance athletes, that they tend to experience increased intestinal permeability, severe gastrointestinal distress, and even low-grade endotoxemia. So that's sort of like the level. If sepsis is a level 10, endotoxemia can be maybe like a level 1 or 2. So it's still not ideal. And again, the interventions recommended there are maintaining euhydration, having a carbohydrate-containing beverage during exercise, probably limiting and timing fermentable fibers, away from exercise, and trying not to exercise in extreme heat. And then just realizing if you're one of if you're part of a population that might be at greater risk, if you are female, and if you have a history of gastrointestinal distress related to exercise, or if you have something like IBS, that you may be more likely to experience some of that gastrointestinal distress. But once again, and that's an area where probiotics really don't seem to help much at all, and they may actually exacerbate symptoms, especially in females. So really, it's like you said, it's not super sexy stuff. It just comes down to diet, physical activity, maintaining proper hydration levels, fueling properly during exercise, trying to be prudent about when you're exercising. And then there really is a dose and magnitude dependent response. So making sure that you're, prioritizing rest as well, and that you're not just running for the sake of running, that you're doing it, if it's going to be longer than an hour or two, there probably should be good reason for that.
You hear that, John? If you're going to run for an hour like you always do.
So I've started Brazilian Jiu-Jitsu back up, and there's like a 10-minute warm-up where you have to like run and do a little. I nearly died.
That means you're in good powerlifting shape.
Hey, it's going to carry me through a meet. And look, there's not a lot of this research done on strength athletes, but I am going to go to them and be like, you need to do your cardio so your gut health can be better.
Mm-hmm. Yeah.
Done. Then you will die in 15 years.
So, because we have the knowledge of this sort of dose-dependent and magnitude-dependent response of gastrointestinal distress being above 70% VO2 max, especially while running, I should point that out. That seems to be the most exacerbatory for longer than an hour, especially in, with heat stress or dehydration, we have that sort of cutoff, but we also have some other important and, potentially beneficial cutoffs, I'll say. So if we're looking at exercise that's lasting less than an hour and it's about 60% VO2 max, we can still obtain cardiovascular benefits without that increased risk of gastrointestinal distress. And in fact, that type of exercise, that sort of moderate intensity exercise, can actually enhance GI motility. So if a person is feeling a little bit constipated, then they might be able to just go out and have, a leisurely, relaxing bout of endurance exercise for 30 minutes. And that could alleviate some of the constipation. And it also keeps it very attainable for us. So, if we do want to institute sort of a whole regimen of, gut health support, even though I use gut health very carefully because it's such an abused term, but, things like eating vegetables regularly, making sure that we have plenty of micro-accessible carbohydrates and fiber, engaging in regular physical activity, even if we're looking at, one study looked at women who were meeting the World Health Organization recommendations for exercise or not. So sedentary versus lightly active versus highly active, just meeting those recommendations seems to increase it seems to correlate with increased microbial diversity and butyrate production. So it keeps it very accessible, very obtainable, that we can do these things and we don't have to buy super expensive supplement or go through some sort of special gut health protocol. You can just eat a lot of vegetables, get plenty of fiber, and engage in physical activity. And those seem to be that's the cornerstone of, gastrointestinal health. And as we start to exceed those things, if we are an ultra endurance athlete, the guys and I were just saying, you have to assume risk there. You realize that you may have some gastrointestinal symptoms and there may be some actual damage occurring to the gastrointestinal tract as a result of that, really hardcore, vigorous, long-term endurance exercise, and then being very careful about, including some of the things that we can do to mitigate that.
Any data on sleep deprivation and the gut?
So they have done a little bit in rodents looking at sort of circadian rhythm disruption, but they haven't really been able to replicate that in humans. But what's interesting is that there's a sort of a circadian rhythmicity to bacterial growth in the gut. So during the day, we tend to see an increase in Firmicutes, which is not so surprising because a lot of the lactobacilli that's part of the Firmicutes group are in the small intestine. And so that's when we have nutrients being delivered to the small intestine, and they can use some of those that they can, grab before we get to them. And then during times of fasting overnight, we see increases of Bacteroidetes. And that also makes sense because we're going to see more nutrient delivery to the large intestine. But some of that could also be just because we have so many more bacteria in the large intestine than we do the small intestine. That we're looking at that's just going to be so much. It's going to be a magnified change, so to speak. So that lends itself to the idea of, some credence to chrononutrition and the benefits of a normal sleep-wake cycle, being awake during the daylight hours, being asleep at night, because it could, correlate with what we see with circadian disruption in humans. So sleep deprivation obviously leads to reduced performance, immunosuppression, dysregulated appetite, and the gastrointestinal tract and the microbiome both as sort of independent of one another. We have, hormones released from gastrointestinal cells, and we also have some interplay there between the microbiome and the nervous system, that they all interact to sort of regulate and influence appetite and hunger. So I would say that, that probably is going to be coming out in the next couple of years. I'm sure we'll learn a lot more about the effects of sleep deprivation. But what we're seeing right now is just sort of, people are looking like intermittent fasting and things like that. And that's another area there where we don't have very much data, but at least we've been able to identify like, hey, we have this sort of rhythmicity and bacterial growth. We have to keep that in mind when we're doing these fasting studies, because some of it could just be normal rhythmicity and bacterial growth having nothing to do with our fasting protocol.
Yeah.
A lot of moving parts.
Yeah.
Yes.
Pretty complex, this microbiome thing.
Yeah.
People love to simplify it on the internets, but I don't think that's really possible.
It's almost like there's nuance or something.
I know. That's what I was saying. Like, there's context and nuance. Like, it's hard for me to give like definitive answers without a lot of other stuff. It's like, well, here's what I'm saying, but here's also why I'm saying it. And, and each of these areas is its own kind of like discrete area of study. I looked at skeletal muscle metabolism. That's really like the focus of my dissertation, microbiome, skeletal muscle metabolism. But you can look at hepatic, you can look at adipose, you can look at nervous system. Like, there are so many, immune system, so many different areas, which is why I'm like, I can't think of it. Like, I'm not an expert because like there's too much to know.
Well, and you gave the example of creatine, and now you have creatine, which is this inert substance. And even then, its effect on the body is extremely dynamic. You have many factors that go into how you're going to respond and what subsystems are going to respond. But now we're talking about an entire ecosystem in and of itself, interacting with itself, then how that's going to interact, and to the trillionth degree where we're talking about microbes. My mind was just blowing when you were describing these things, because I don't know how you study it.
I study just like this much of it.
Well, and the cool thing that I think blew my mind is when you're talking to the summit, talking about some of this bacteria kind of taking over some of the duties that we would normally see our body doing, RNA transcription, DNA transcription, and proliferating certain processes in the body, and all of a sudden you realize there's a lot going on here. And when you see a clickbaity title, you have to look at it and be like, it's probably more than just that. This isn't the panacea people are kind of making it out to be, because it's just so young when it comes to the amount of data.
Mm-hmm. Yeah, I think in the past, Patrice Connie, he's one of sort of the founding fathers of this literature. And one of the first names that I remembering because his lab coined the term metabolic endotoxemia, which was sort of the crux of my whole dissertation proposal. But he recently did sort of just an editorial piece on the hopes, threats, and promises of the gut microbiome, saying like, hey, let's not apply causation where there's only correlation. And I want to say that it was in the past 5 or so years that we've seen like 80% of the publications of the microbiome come out, because it's been an absolutely exponential increase. I mean, when I finished, no one knew what the microbiome was. No one cared. I didn't have anyone to talk about my project with. And now it's just like this huge hot topic. It's really interesting to see. It's got that snowball effect going now.
Yeah. Social media will do that to some things too. Mm-hmm. Like you said, as soon as somebody links it with weight loss, whew.
Oh yeah. Yep. That fecal microbiome transplant. Yeah, exactly. Like that was going to be the next thing, like they did that in mice, and it was like, oh, well, we can make lean mice obese and vice versa. And, this is going to happen in humans. And guess what? They've done that in humans. And guess what? It didn't do anything. Then it's not, no poop transplants for us.
I can start like getting rid of my fit friends poop that I've been collecting.
Yeah. Yeah, if you've been saving that in your freezer, just, you can donate it or something. Like, yeah, it's not going to do you any good, unfortunately. Or like, there's going to be probiotics now. It's going to be like, well, I don't know, like the LeBron James like probiotic, and it's just like they sequenced his microbiome, and now, yes, exactly. Oh yeah, I remember being asked about that. Like, do you think we're going to have designer microbiomes? Like, oh, I don't know, maybe like much later. But it's a lot. It's like when we were looking at like genomics stuff, like, oh, that's going to be the next thing. We're going to do like personalized nutrition based on your genes. And then we're like, oh, wait, actually, your microbiome is a better predictor of some things than your genes. And that actually does appear to be true. So that's kind of interesting because it's like we just we try to like simplify ourselves too much, and like, oh, here's this button I can push and I get an output. And it's like, no, we're complex systems. That's not how this works.
I'm not over the designer probiotic. Lactobacillus by Gucci.
Yeah.
All right. So any closing thoughts before we sign off here?
Oh, for me?
Yeah, for you.
I was waiting for you guys to ask me another question. I would just say, I want to encourage people to sort of ignore the current message of you being broken in some way. Like we need to heal the gut or reset the gut or detox the gut. That alone, it is such a moneymaker, and it is really an insidious message that I think seeps its way into a lot of different, not just in the area of gut health, but in the area of, weight loss and fitness and things like that. We have this sort of, no pain, no gain thing. And like, if there's not a problem, I'll make one so that I can sell you something to fix it. And it really does consistently come back to the basics when we're talking about gut health and most other things, making sure that your nutrition and training and sleep and stress management are all in order. That's really probably the best you can do. And if you want to do it, like I said, from a gut health perspective, great. And then you'll accidentally be eating a super nutritious diet. And that's, I think it was when I was on with, Greg Nuckols and Eric Trexler, I think he said he that's what he used to tell people when they're like, oh, nitrates, like, oh, that's so exciting. Should I be supplementing? And he's like, well, lots of vegetables are high in them. So you could just eat lots of vegetables and that would be good for you also.
It's boring.
I know, right? It's not sexy enough. People want to know like what special supplements they should be taking and whatnot. And it's like, no, there are, for most people, it's just the basics. There are certainly, special considerations and things like that. Like we talked about with the ultra endurance athletes where, an extra layer of intervention might be beneficial for them. But, start at the bottom first and realize that there's very, very little you can probably change about the gut microbiome. And people will try to sell you a lot of things, contrary to that evidence. But, save your money on the vegetables and, gym membership or whatever you need to do to be physically active, and like maybe a massage or something to help you manage stress.
Awesome. Where can people connect with you if they want to find you?
They can find me on Facebook and Instagram at Vitamin PhD. I have a website, vitaminphdnutrition.com, where they can look at my other podcasts, other things that I've posted some videos on the microbiome, lectures in a few different places, RP Plus, Revive Stronger Experts, the Muscle and Diet MBAs both have lectures on the gut microbiome. So absolutely check those out. And feel free to shoot me a message through that website or DM me on Instagram. They can also find me on renaissanceperiodization.com if they like to do email type coaching. And I also do video coaching as well. So kind of two different veins depending on what you're looking for. But yeah, absolutely reach out and say hi. I try to answer every DM. I think unless I like accidentally delete them, I answer them. That's awesome. Yeah. Well, thank you so much for coming on. Quinn, Jared, thank you guys so much.
Gabby, this was great. Thank you.
Yeah. Thank you.
Glad. Thank you guys. And it was great talking to you.
We'd like to thank Dr. Gabrielle Fundaro for coming on the show. You can check out the show notes for links to Dr. Fundaro's contact info. We highly recommend following her work. And of course, thank you to my homies, Jared Maynard and John Flagg, for steering the ship alongside me. And thank you, the clinical athlete community, all 6 of you, for joining us on this journey of knowledge and improved practice in both the gym and clinic. If you want to dive even deeper into the clinical athlete community, you can check out all that the Clinical Athlete Forum has to offer, which includes our Clinical Athlete Academy courses, amazing discussions and networking with professional clinicians and coaches, as well as students, and just our overall hub of knowledge in regards to athlete health and performance. And don't forget to check out details to the CALU Summit coming up in September. Thanks, everyone, and talk to you soon.